We will now turn to the final Sarah, Sarah Rugheimer, who will be talking about life as we do know it. Sarah, please enlighten us.
So I'm very excited to be here, and I wanted to start with a question to all of you. So who thinks that we will have a robust detection of life either in the solar system or on an exoplanet within 50 years? Raise your hand. Keep those raised if you think within 30 years. Keep them raised if you think 20. 10? Who thinks we already have it? All right. So I wanted to, whenever I think about finding life on other planets, I'm pretty sure this is the picture that the public has when I give an outreach talk or speak to the media. What I'm really claiming as a theorist is that we're going to find a beautiful spectra, no error bars on this spectra, with clear signs of oxygen, methane, vegetation, red edge, water. What we're going to find, pale blue pixel, lots of error bars in the observations, maybe multiple models fits the observations, and we're going to argue about it for a long time. And so when we think about what we can know about the Earth and the solar system planets, we have like petabytes of data, right, for our own solar system. And what we know about exoplanets is we know their mass, their radius to maybe within 20%, maybe, you know? So when we think about the solar system is a wonderful laboratory for us to really get into details on planetary processes, and exoplanets provide that statistical context for understanding our solar system in the universe. But I want to make this point that the habitability and the habitable zone really have nothing to do with the theoretical limits of life. I see this conflation all the time. So just because a planet is in the habitable zone doesn't mean it's habitable. Just because a planet is habitable doesn't mean it has life. But it's a really useful process for us to help target select when we're trying to think about where could we best remotely detect life on another planet, because we have to be able to look for something with a large surface biosphere that has an atmosphere that it's interacting with. We just can't detect life in Europa if it's an exoplanet. You know, there's no atmosphere for us to look at. So that's just one thing that I think gets often mistaken. So I like to frame this as we have two challenges. We have these technological hurdles that we just heard about in the last talk, very difficult. And then we also have these interpretation hurdles. What does it mean when we find these molecules? Are they life or are they not? And that's very difficult. So I like thinking about this, this quote by Feynman's a wonderful quote. Science is a way of trying not to fool yourself. The first principle is that you must not fool yourself, and you are the easiest person to fool. And it's true for us scientists as well, right? So we have this famous Cydonia region on Mars, this famous face on Mars that was taken by Viking. And then with higher resolution data, the face goes away, right? This one always gets me. These balls are all the same color. They are. I've even proven it to myself with like an RGB color picker on my computer and gone and made sure that they are indeed the same. But we just can't see it that way because our eye is expecting to see something else. And so I want you to kind of keep this in the back of your mind when we're thinking about our interpretation of finding life on other worlds. So this data is going to be very difficult to interpret. It's a pale blue pixel. And there's really only one type of detection that I would say will be unambiguous, and that's this sort of, hello, here we are. Then we'll know, that we found life. So there's two ways of searching for biosignatures and from microbial life at any rate. We can look for life as we know it. This is for looking for signatures based on carbon-based biochemistry, oxygen, ozone, methane, with water and CO2. And we can think of our own Earth spectrum here. Sarah already introduced you to this wide difference in photons we get from the sun compared to the Earth. We can look for signs of oxygen, methane, vegetation, red edge, ozone, and these features, and be able to have a good sense of how we would detect an Earth around another star. We can also look for life as we don't know it. And there's some really great work by Sarah Seeger's group saying, okay, let's consider all the possible molecules that life does produce on Earth. Which ones are actually stable and volatile in the planetary atmosphere? Which ones don't have a geological false positive and that we could detect? And they came up with over 16,000 molecules. But sadly, we don't have the spectra for most of those molecules. We only have the spectra of inequality for about 0.04%, which means we just couldn't detect them. Like if we see those molecules in the spectra, we don't have the theoretical background yet to detect them. The main biosignatures on Earth, of course, are this combination of oxygen or ozone in combination with methane. But you could get these without life. You can get oxygen or ozone by UV photolysis of water or CO2. You can get methane from serpentinization or volcanoes. So individually, these are not good signs of life. On Earth, biology is producing large quantities of both of these gases, but if biology were to go away, the molecules would go away. So it's really this combination on Earth, this disequilibrium of an oxidizing plus a reducing gas that gives us the strongest indicator of life on our planet. And that's the strongest biosignature still to date on exoplanets. And I want you to note it's always a combination of gases. There's not a single gas that's going to be a good biosignature, and I think that's going to remain true. Also, there's Archean disequilibrium biosignatures. So methane plus carbon dioxide and water without carbon monoxide. Why? Because carbon monoxide, if the methane was coming from volcanoes, you'd also be producing that carbon monoxide. So this has been proposed as another disequilibrium pair that we might be able to detect, say, an earlier phase of life evolution on another planet. We could look for these alternative biosignatures when we're thinking about these interpretations, and there's a lot of reasons to want to do that. It's not Earth-centric. Great reason. The cons are we have no idea if those biosignatures exist or if alternative biochemistries exist at this point, though of course we're making some good headway. We can't tell if there's life in our own backyard, here in our solar system, on Titan, on Venus, Mars, Europa. And so, we need to, I think, do this exploration in the solar system. We need to go to Venus. We need to go to Titan. Look for these sorts of different biochemistries, see what complex chemistries are there, and really use that as our test bed for then how we are going to explore in the rest of the universe. Same with Mars and Europa, more classically Earth-like, habitable, but still great places for us to go. We could look for unique biosignatures, things like methyl chloride, chlorofluorocarbons, dimethyl sulfide, phosphine. There are fewer known false positives, still some false positives for these, but they are, let's see if this slide advances. There we go. They're usually rare and limited to a single microbial species, an evolutionary quirk. So that means they're only produced typically in small quantities. And, for the other molecules out there, the other 16,000, we just don't have a lot of spectra for them to look. So when we go back to thinking about Earth biosignatures, there's some pros to this. One is that water and CO2 are abundant in the universe. They're everywhere we look. And carbon's more abundant than silicon. It forms more complex chemistry. Water is polar and has these unique properties, which seems to be really advantageous. I also often get this question, and so I wanted to address this specifically. Why are we obsessed with oxygen? And I think it's worth mentioning that it's because oxygenic photosynthesis uses water, CO2, and photons. These are abundant on Earth and in the universe, and these three are used for oxygenic photosynthesis. And it's because they're so abundant that is why oxygenic photosynthesis has been the single most successful biomass building product on Earth. So that is why oxygen is useful as a biosignature. Selfishly, complex life loves oxygen because you get a lot of energy for it. And so all complex life uses oxygen for respiration, but that's not why it's a biosignature. That's not why we care about it as an exoplanet. Aerobic respiration does give the most amount of energy, though, in your redox pairs, and that is why complex life uses it. So, oxygenic production, why it's a biosignature, it's using these three abundant common constituents for microbial life. It's interesting to us for complex life because it gives a wide excess of energy that allows us to think, move, do exoplanet research, and ponder these questions. So when we go back to searching for Earth-like life, the main pro is, of course, we might be able to better understand what we see. Oxygenic photosynthesis uses these common constituents, and there's no shortage of places to look. It's estimated, say, 40 billion Earth-like planets in our own Milky Way. The cons is that these classic biosignatures all have strong geological or photochemical false positives. Other things produce these molecules. The false positives are strongest for the most common star, for M stars. And so we need multiple gas detection, potentially invisible and infrared. It makes it much more expensive. And still we could be fooled. Think back to, the Cydonia region on Mars. When data is fuzzy, we can interpret a lot into it. And so we can't visit the planet and see alien kangaroos jumping around on the surface and really prove to us that we have found alien life. This pre-luminous main sequence M-dwarf false positive scenario I'll just run through because a lot of the scenarios look like this. Basically, you have strong amounts of extreme ultraviolet light, breaks apart the water into hydrogen and oxygen. Hydrogen is light and escapes. O2 atmosphere is left behind. The planet is dry and it's lost its water, and the amount of oxygen will depend on how much reacts with the surface, fundamentally. We also have strong flares from the M stars for their whole lifetime. That's just Earth to scale. And this movie shows you the strongest flare from the sun, X45. You'll see it right about now. Oh, there it is. Strongest flare. Now look at the M star flare. That's the strongest flare we've observed on an M dwarf. This is why it's a problem, folks, So this has given rise to this concept called the cosmic shoreline. So we have this idea that, an atmosphere just might not be possible. A thin Earth-like atmosphere in the habitable zone around an M star might just not be possible, and it depends on the amount of total XUV irradiation that's happened over the course of its lifetime and how massive the planet is. All right, so let's wade into some controversy. Let's look at some recent biosignature claims. So is there phosphine on Venus? To be fair, they didn't claim that this is necessarily life on Venus. They're saying it's either an unknown chemistry, possibly life production. Certainly why it's interesting is phosphine is produced by life on Earth, and it's only produced by life on Earth. And in terrestrial planets, that seems to be the best way of producing it. We can produce phosphine abiotically in Jupiter or Saturn, but in a terrestrial planet, phosphine seems like a great biosignature. So Jane Grief, Sarah Seeger, and their teams, said that maybe we have found phosphine. They claimed a 15 sigma significance in that first detection paper, going back to sigma. So like say a 3 sigma result has a 99.7% chance of being true. So 15 sigma is quite a strong claim. There's always 2 checks we need to make when we're thinking about biosignature detections. Is it the molecule? Have we actually detected the molecule? And then is it life? These are the 2 questions that I think we always need to ask ourselves. So of course people did. And there was some pushback on that initial discovery paper from Ignace Snellen's group. And here you can see that we have phosphine in the top left, and then regions of the spectra where there are no molecules. This is before modeling the instrument systematics, and then after modeling instrument systematics, this is where you see the phosphine line, and then you can see signatures, features pop out where there's nothing in the spectra. So they argued that after we need to be a little more cautious when we're modeling our instrument systematics, use a gentler approach, and then they claimed that the phosphine was detected with around a 2 sigma significance. Well, the debate rages. There are papers back and forth. Let's reanalyze the Pioneer data. Let's do another analysis of the ALMA data. So there's lots of work. This is what I love about scientific controversies, is that it generates so much cool science. We learn so much by people making these claims. It's wonderful. It's part of the scientific process. And so we've seen a flurry of papers going back and forth, and I really recommend, if you want to keep up on this, Sarah Seeger's creating a good document of all of the back and forth work and what each claim is making, so you can follow that progress. We'll figure it out eventually. What I love about phosphine on Venus is we can go there. we can go, we can figure out, is there phosphine? Is it from life? We're going back to Venus, which is very exciting. So there's missions that are scheduled to launch soon, even as soon as next year. And we're hopefully going to put to rest this phosphine question. I recommend also Clara Sousa Silva talk on just this process of vetting biosignatures. She does a good job of going through some of those complexities. Dimethyl sulfide. I'm not going to dive into this too much because we have some talks coming up later about it, but needless to say, it's been in the news, and we always have to make these two checks. Is it the molecule? Is it life? So there's been laboratory studies of viability of life in H2 atmospheres that have shown that microbes can be active and produce biosignatures. Indeed, they can produce dimethyl sulfide. This is from one of Sarah Seeger's paper. We've also seen research last year that shows we can produce dimethyl sulfide without life, especially when there's methane around. And also we've seen evidence of dimethyl sulfide in a comet. So that I'm going to leave the rest of that controversy for later today. I wanted to briefly touch on what about AI? There's lots of cool stuff we can do with AI to see the differences that life imprints. This is a paper looking for molecular biosignatures in old Earth rocks, but that life just leaves a different imprint and it looks different. And so we might be able to, this research showed that it kind of clusters, the molecular imprints it leaves clusters, and we might be able to use this to better analyze data, and I think eventually we hopefully will be able to do something similar for exoplanets. With my remaining time, I wanted to briefly touch on what's next. Who's heard of the LIFE mission? One of my favorite missions, the Large Interferometer for Exoplanets. Astronomers are not usually known for their acronyms, but we did a good job on this one. So our goal is to obtain the thermal emission spectra of an appreciable number of habitable exoplanets in the habitable zone, rocky planets. It's using nulling interferometry, and we wanted to be able to find 30 to 50 or so of these, such that even a null result, even if we don't find any Earth-like biosignatures, that would be interesting. So that's the drive. Nulling interferometry, basically we can use a phase inversion in our beam when we combine the light and null out the starlight and then be able to directly get photons from the planet. That's the idea. It's a very complementary view to NASA's habitable worlds, so we're able to get some different molecules. Habitable worlds is able to detect O2, which life wouldn't be able to do, but we are able to get CO2 and methane, as well as the surface constraint on the surface temperature, which is really important for the habitable context of this planet. But even something like detecting oceans is really difficult, even with a wonderful future mission like LIFE. And so there's some work that I've been leading on this of the running retrievals on different water abundances with different assumptions. And needless to say, it's just hard to constrain the abundance of water unless it's kind of around Earth-like levels. And I don't have enough time to fully go through this, so I'm just going to skip ahead just to say that hopefully the water vapor in the atmosphere is enough of an indicator of surface water, though. So I wanted to end that, we're on this journey. We are hoping to be able to detect Earth-like signs of life on other planets. Maybe we'll find some other molecules, other interesting biosignatures. Certainly we'll see things that we didn't expect on those planets. And, I want to leave you with this thought. So even if we're not alone, we'll be satisfied. I've spent this whole 18 minutes talking about finding microbial life on another planet. Is that enough? Even if we find complex life, like a ladybug, or you say ladybird, I think here in the UK, that's a very complex organism. Would that be enough? We can't explain, our existential despair to a ladybug. And so do we really need that intelligent life signal, to feel like we're not alone? And so I leave you with the question of, even if we're not alone, will we still be lonely? Thank you very much.